FRQ number 2. Let's start with proteins are essential for almost all aspects of life, but using location, structure, and function describe the important uh, proteins into the following three systems: cell cycle and division, B, cell movement, C, is signal transduction and communication. With A, cell cycle and division, actually, this is three questions in one. They want you to talk about number one, they want you to talk about protein in cell division. Then they want you to talk about protein used in mitosis. Then they want you to talk about protein used in DNA replication. So let's start with the first one. Protein that controls cell division, they want you to talk about histones, which is positively charged proteins in which DNA is bound to. DNA is bound to histones. They want you to talk about growth factors and hormones in terms of signaling from cell to cell and how signaling to for cell division. They also want you to talk about proto-oncogenes products, which works as a braking system or an accelerator. Whenever we need to go forward, they just accelerate. When we need to stop, the proto-oncogenes put a brake system. That's it. They want you to talk about cyclin dependence kinase, CDKs. Of course, CDKs remain constant throughout the cycle, but they will be activated by cyclin. So CDKs, which is cyclin dependence kinase, when they bind to cyclin, they send the message for the cell to divide. When CDK and cyclin combines, we form a maturation promoting factors, MPF, and this is how we continue. And then they're going to talk about proteins used in mitosis. Centrioles are arranged in 9 plus 2. We know what 9 plus 2, 9 triplets, microtubules in a ring that have microtubules between them. Talk about microtubules when the spindle fibers of mitosis attached to kinetochores and used for cell elongation during anaphase. They want you to talk about centrosome, which is the microtubule organizing center. Then they want you to talk about kinetochores used to move chromosomes to the midline at metaphase. The third one, which is the protein used in DNA replication, let's start with helicase. Helicase divide the double helix for replication. Then comes primase, begin replication with RNA. Then which will be preparing for DNA polymerase, because when DNA polymerase comes, it will start elongation and replacement of RNA primase or primers. Talk about nucleases which they cut DNA mismatch and other mistakes. And finally talk about ligase link sections of DNA. All this for A, cell cycle and division. Now let's move to B, cell movement. Cell movement, again, talk about microfilament, pick actin, and the microfilament we have actin and myosin. Just talk about actin and say the thinnest filament bound to plasma membrane and used for contraction. Talk about integrins, proteins that span the plasma membrane, collagen, which is abundant protein attached to integrin for the extracellular activity. Also talk about myosin. Myosin is a large protein used for, cytoplasmic streaming. Talk about again flagella and cilia. Flagella. It's 9 + 2, 9 doublets of microtubules in a ring around 2 single microtubules. And I think that's it for cell movement. Let's go to C, signal transduction and communication. Okay. Let's start with first messenger, peptide hormones that acts on a specific target tissue. We talk about GPCR protein receptors, GPCR protein receptors. Receptors, a protein embedded in plasma membrane that receives hormones. Okay, now they have relay protein embedded in plasma membrane, which stimulate or inhibit G proteins. Now, inside the cell, we have effectors protein acted, causes changes, which is, we call it the second message or messenger. Example is like adenyl cyclase, this is for the protein receptors, the GPCR protein. When it comes to steroid hormones are different. Steroids receptors found in the nucleus. They receive steroids hormones and bind with DNA and cause regulatory protein to bind with DNA. So remember, steroid receptors are different than GPCR protein receptors. Various protein kinase and phosphorylarases acted on by second messenger, sometimes third messengers, whether activate or deactivate reactions. Of course, they amplify the first message. One more point, maybe talk about structural protein like opsins, OPSINS, which is initiate many sensory responses. And talk about ion channels. The ion channels are responsible for neuronal functions. And that's it for question number 1. Let's move to question 2. Since Watson and Crick's elucidation of the structure of DNA, many scientists have sought to concentrate their research efforts on DNA, the code for life. Taxonomists and phylogeneticists use changes in DNA to study classification and evolution. Describe A, all types of DNA within prokaryotes and eukaryotic cells. B, the sources of variability in DNA sequence and chromosome number. C, how scientists have studied changes or differences in DNA using the theme of science as process. Let's talk about the one we always mentioned probably in the previous FRQ, which is types of DNA within the cells. When it comes to prokaryotes, bacteria, DNA without a nucleus bound to plasma membrane, not associated with histones, okay? And it's circular. When we talk about plasmids in prokaryotes, circular piece of DNA that can be exchanged by prokaryotes, okay? That's it, I believe. For the eukaryotes, DNA, DNA within nucleus, okay, bound to histones, arranged in chromosomes. They want you to add other stuff. They talk about the DNA in the mitochondrial DNA. The mitochondrial DNA, which is like a prokaryotic-like DNA, of course, the mitochondrial DNA reproduces on its own. not only the mitochondrial DNA, but also the chloroplast DNA. If you forgot, go back to the endosymbiotic theory. The endosymbiotic theory, they talk about how billions of years ago, one large prokaryote engulfed another smaller prokaryotes, and the smaller prokaryotes became evolve as mitochondria and chloroplast. Uh, then talk about the differences in DNA and types of changes. Okay, so we are still in 2A. So 2B, the differences in DNA and types of changes. And they want you to talk about mutation. Mutation is the major cause and source of most variation. Okay, where do you know that? Talk about point mutation, change in a single nucleotide. Talk about silent mutation, produces no change. Talk about missense, produce changes in amino acids coded for, which means with the missense, we still bring the same amino acids, so no changes. Talk about nonsense. Nonsense is like when you get UAA, which is a stop codon. That's it. We put a braking system, so everything stops. Talk about insertion or deletion, which is the most damaging. This is, we call it frame shift mutation. Talk about inversions, which is the reverse in order. Translocation, moving DNA from one chromosome to another chromosome. I believe this is it. Transposon, transposon, pieces of DNA that move around, they will move from chromosome 1, maybe you will find them going to chromosome number 10. So this is transposon. How scientists have studied changes or differences in DNA using the theme of science as a process. C. Talk about gel electrophoresis, which is an electric current causes the negative charged DNA. DNA has phosphate group, negatively charged. So the negatively charged DNA move towards the positive pole. Then the separation of DNA in agarose gel is based on size. The smaller the molecules, we call them beads or DNA fragments, they move faster. Then talk about restriction enzymes. Nucleases that cut DNA usually at palindromic sites. What's palindromic? In English, when you have a palindromic word, it's like you read it from left to right or right to left, same thing. Like race car, both ways, or kayak, or some students say wow. So this is palindromic. In DNA, you have ACGC, for example, in one strand. When you go from the other strand, ACGC, same thing. So this is palindromic. You read it from top, bottom, same thing. This is palindromic. Okay, of course, subjecting different DNAs to the same restriction enzyme, and then using gel electrophoresis. Talk about how scientists, they can find if there's a difference, or they divide the genome into smaller sizes. How? We use RFLPs. What's RFLPs? Restriction, fragment, length, polymorphism. In this way, we have a DNA. DNA is digested with restriction enzymes. Then this can be used as a DNA fingerprint. Okay, using Southern blot. Southern blot is the first DNA techniques used back in 1986. Southern blot. Then scientists, they probe the DNA with a radioactive single-stranded. They bring a single-stranded DNA, radioactive, or it could be nowadays no radioactivity because it's dangerous. They use fluorescent. Okay, and they allow it to attach complementary to the regions. This way, they can determine gene location. It's determined for DNA fingerprinting. They can do gene mapping. They can find out whether we have, um, for cystic fibrosis, for example, whether we have cystic fibrosis on the genes or not. They can do probing. Uh, PCR is also used, polymerase chain reaction. Of course, we use different temperature. Why do we use PCR? To amplify smaller fragments of DNA. Smaller amounts of DNA can be amplified. If it's too small, we cannot work with DNA, so we have to amplify DNA, and then we use restriction enzyme. So if we have a smaller amount of DNA, we have to amplify it using polymerase chain reaction, PCR. How do we do it? We subject the DNA to different temperature cycles, heat, then cool down, then heat again. Okay. This sequencing determining the actual base sequence. Okay. Of course, we can use fluorescent labeled, nucleotides, and we can use cDNA, which is complementary DNA. Okay. Question number 3. In order for plants to become the numerous and large life forms they are on Earth today, they require several adaptations from their early beginning. Describe these adaptations and how they help plants make their move. Not, it's an okay question. Okay, yes. Talk about fine root hairs, which is increase the surface area and therefore absorption. Okay, makes sense. Talk about roots, where they have association with fungi or mycorrhizae to increase the water absorption. Then cuticle on leaf surface reduces water loss. Okay. Talk about guard cells, they control the stomatal opening. One thing about C3 and C4 plant, talk about C4 plant, which is plant that allows stomata to be closed longer. When they are closed longer, they can survive in dry conditions. Of course, how do they maintain this? Because they have increased carbon dioxide assimilation, they have efficiency. Cactus, is one of them. What else? Talk about xylem is the vascular tissue in plants. And that's it. Reproduction. Gamete transfer, that's one. Sperm swim to ovules or embryo sac in few plants. Ferns is one of them, where the sperm swim to ovules. Imagine, the ovules are the